Hello, everyone. I would like to welcome you to today's ESDR Kitchen, Sweet and Sour. My name is Christina, and Remco van Doorn is my co-chair for today. We are actually very excited to have two exceptional speakers today, Christoph Ellebrecht from the US and Ricardo Grieshaber-Bouyer from Germany. And it's my honor to introduce Christoph first, who will give an around 10-minute presentation on his work on CAR T cells in autoimmunity. And then Remco will continue with our next special guest, Ricardo. So Christoph is actually German. He studied medicine in Lübeck, where he also started his clinical residency in dermatology. But then he went to the US to do his postdoc, already on the topic of very precise and specialized immunotherapies with CAR T cells in the setting of autoimmune diseases, with a special focus, in fact, on blistering diseases like pemphigus. And then he continued his clinical residency training in the US at the university at UPenn, where he now also has his own lab and where he's also working as an assistant professor. So it's really a great honor to have you, Christoph, today as our speaker. And if you're ready, I would suggest that you start your presentation.
Oh, wonderful. Thank you so much for this kind introduction, and thank you for having me. Can you continue to share? Yes. Can you guys see my slides okay? Wonderful. Okay, yeah, thank you so much for inviting me to talk about CAR T cells and autoimmunity. It's been a rapidly expanding field and quite exciting to see what's happened over the last couple of years, 5 to 10 years. With that, let's start some disclosures and just to give you an introduction what CAR T cells are or what a chimeric antigen receptor is. More than 2,000 years ago, people already were fascinated by the concept of merging new things, taking known elements and putting them together into something new. And that's basically shown here with this chimeric creature with a lion head or another head of a goat, elements of a snake. And this really has fascinated people for centuries. And that's basically what we do when we make CAR T cells. We take existing receptors and merge them into new receptors. We take parts of the B cell receptor shown here, mostly the antigen binding part, and then also parts of the T cell receptor, and mostly the stimulatory signaling domains and co-stimulatory domains that provide signal 1 and signal 2, basically stimulation and co-stimulation for survival and functional plasticity. And when you merge these two receptors, you basically end up with a chimeric antigen receptor or CAR. So again, it's basically the antigen binding domain derived from a B cell receptor from the surface immunoglobulin that directs the CAR against an antigen of choice. It can be a cancer antigen or something that is specific to autoantibodies or autoreactive B cells. And then you have these hinged transmembrane co-stim and usually CD3 derived domains all from the T cell. And that's why it's called a chimeric antigen receptor. When was the first CAR T cell described or the first CAR described? People think this is a brand new novel concept. It was actually quite a while ago, a couple of decades ago, almost 37 years ago, that this was described in a really nice paper by a group from Japan. And in this paper, they basically described the expression of a chimeric receptor composed of immunoglobulin-derived V regions, variable regions, and T cell receptor-derived C constant regions, which is basically exactly what I just mentioned. And they speculated that in the future, in the discussion of their paper, it might become possible for T cells to recognize any antigen without MHC restriction to be produced by this technique described in this paper, which was it's quite fascinating that they actually predicted what they could do with this technology and really took off. It's not, when we look at CAR T cells today, it's not just a chimeric receptor, it's also a chimeric field. If we look at the history of what had to happen since 1987 here on top, CAR technology had to evolve. We had to have understanding from adoptive cell therapy, bone marrow transplants, in order to actually further this field. And then at the bottom, you see cloning, gene therapy, and understanding of viral vectors was very important. So all of this had to come together. And in the beginning, none of these fields were actually envisioning, I assume, that they would end up with an FDA-approved CAR T cell product. So it was initially, as I mentioned, immune receptors, cancer, and immune system. That interaction was studied or started to understood over 100 years ago. And then gene engineering, genome engineering started off in the 70s. Learning from HIV, studying a virus actually helped us make these lentiviral vectors that we use in the clinic today. And then in the beginning, people wanted to use it for cancer, then for treating HIV infection, and then also in autoimmunity. The first CD19 CAR that we will probably hear about in the second talk more was basically described in 2003, and the first report of clinical efficacy was in 2011, roughly, and at that time in their patent application, this was done by Carl June's group here at Penn. They basically speculated that this could also be used in autoimmune disease, and we basically published the first AA CAR construct for autoimmune disease treatment in 2016, and then the cancer-approved CARs started taking off in 2017. So this is really a chimeric field. Why is, for example, CD19 an ideal CAR T cell target if you think about autoimmune diseases, and that will likely be discussed in the second talk as well. It is basically an antigen that is expressed on the surface of B cells from very early developmental stages, pro-B cell stage, down to the plasma cells. There's just a very small proportion of plasma cells that don't express CD19, so it's expressed widely. And then other things that we can target, for example, with the chimeric autoantibody receptor, are expressed more at the memory phase and are more restricted to certain B cell developmental stages. BCMA is also a target of CAR T cells in clinical trials and FDA-approved constructs, and that's more restricted to antibody secreting cells, which might be a specific application. How do we make CAR T cells? It's a living drug, so we really use T cells and then engineer them. We remove them. This is how it's currently done. Remove them from the blood of patients to get the T cells. Then introduce a gene by, for example, lentiviral or retroviral transduction. And so that these T cells start expressing the CAR, we expand them in the lab and then reinfuse them into patients where they, if we treat cancer patients, for example, find the cancer and deplete the cancer, or if it's B cells or autoreactive cells, they should find these cells and also deplete them. And this was first done in, as I mentioned, lymphoma and leukemia patients. And this is one of the first reports from Carl June's group here at Penn, where they, for the first time, showed efficacy of CAR T cells when they used a certain costimulatory domain. And on the y-axis in this graph, for example, you see the transgene copy number and then the days after infusion of these CAR T cells. And you see that there's persistence of this transgene in patients' blood, indicating that the CAR T cells persisted. When they looked in the bone marrow aspirates, they saw the same thing, that the transgene persisted. And before infusion, the patient had a lot of CD19 positive kappa chain restricted B cells. And then 1 month after infusion, all these cells were gone. And the patient that persisted for, in this case, several years or over 10 years, this remission. So the patient was basically cured at that time, which was is really impressive. And it's obvious that if you have something that is driven by B cells, you could use this in autoimmune diseases as well. Obviously, because it's a living drug, there are also safety issues that need to be addressed or thought about. Tumor lysis syndrome is something that comes up in cancer patients all the time. You deplete a lot of cells die and release certain markers that can be harmful, can be seen here. And then something that wasn't really known until the CAR T cells started to work, cytokine release syndrome, where a lot of cytokines are released, IL-6, one of the most important ones. And that's why a lot of patients with CRS are being treated with anti-IL-6 nowadays. And then we learned also that there's another syndrome called ICANS, or immune effector cell-associated neurotoxicity syndrome. Starts a little later than the CRS, and then basically patients have neurologic symptoms. It's really not well understood how this works, but oftentimes if it's just mild, it's reversible and just requires supportive care. But these are things to keep in mind. The question is, does that really apply to autoimmunity, and to what extent? And then in cancer patients, factors that we learned are associated with durable remissions. Depth of the initial remission, depleting the target cells is really important to keep patients in remission. Malignancy type, is it a lymphoma? Is it a leukemia? And do you actually achieve a complete response? Tumor burden, how many target cells do you have in the beginning and where are they located? And then importantly, lymphodepleting chemotherapy is very important in cancer patients. Do you to create space in the bone marrow and make sure that the CAR T cells engraft well? And obviously, CAR T cell levels peak. How many do you have at the maximum level? And then how do they persist? And we just published a paper. You can look into that if you're interested in how CAR T cells make the decision whether to become long-lived or short-lived through asymmetric cell division shown here on the right. Lymphodepleting chemotherapy, I want to talk about a little more because this might be important in treating autoimmune disease patients. You see here, the copy number after lymphodepletion is basically very high, goes into over 10,000, 40,000 copies per microgram PBMC DNA. If you don't have lymphodepletion up front, and this is done in cancer patients, you hardly see any engraftment or persistence of these cells. And not only is doing lymphodepletion important in these patients, but it's also the kind of lymphodepletion that you have to use. So if you don't use fludarabine, you don't get this persistence. And that actually in ALL had an outcome difference. So if you use fludarabine, the outcome is important. So it's not just the CAR T cell itself, it's also how you give it, what dose, how do you treat patients before and during expansion that can have an impact on outcomes, at least in cancer patients. And we're still learning how this applies to autoimmune patients. So the conventional wisdom I would say 10 years ago was autoantibodies in autoimmune diseases are obviously important, but we usually distinguish between autoantibody-mediated diseases and autoantibody-associated diseases. And it's a very easy, simple distinction. If you transfer these antibodies to an individual or an animal, a mouse model, the autoantibody-associated diseases usually don't cause a disease, whereas these diseases act, when you do the same experiment, you see this disease, for example, pemphigus vulgaris, you transfer the antibodies, the anti-desmoglein antibodies, and you can induce this condition. So it was 10 years ago obvious, or at least that's where was an original assumption, that the better CAR T cell treatable diseases were the autoantibody-mediated diseases because the B cells are central to disease etiology, which turned out not to be true. Even autoantibody-associated diseases seem to respond very well to CAR T cell therapy. And that's what we assumed. And based on this, in the last 5 to 10 years, there were clinical trials, for example, using an anti-CD19 CAR T cell that depletes all B cells. And we hear about that in the second talk, quite potent, very good results in a number of autoimmune diseases. And then what we also developed, which was the first in human clinical trial for autoimmune disease CAR T cells, the so-called AA CAR, the chimeric autoantibody receptor T cells, which basically give you antigen-specific B cell depletion in theory, where we replace antibody-derived domain with the actually autoantigen to specifically deplete these. While this seems to be generally safe, the efficacy was somewhat muted, and there's no clear clinical benefit in these clinical trials to date. With that, open questions in CAR T cell therapy and autoimmune disease. What is the role of lymphodepletion? Do we need controlled clinical trials? Sorry about that. Lymphodepletion doesn't seem to be sufficient to induce remission in pemphigus in AA CAR trials. So that is interesting that the lymphodepletion itself may not induce remission, complete remission in autoimmune diseases. The mechanism, are B cells indispensable in autoimmune diseases that are associated with autoantibodies? For example, in lupus, are B cells just antigen-presenting cells and the antibodies are just, a marker of the disease? What are biomarkers of responses in these conditions? Do we need the same dose requirements in other autoimmune diseases compared to, for example, lupus or cancer patients? How do we select patients to treat with CAR T cells? can we actually scale this? Can we pay for this? Is it affordable to treat any autoimmune disease patient with CAR T cells? And is it safe? In some patients, at least in cancer patients, there seems to be some sort of oncogenic risk. It's very, very low. We still are trying to understand what is actually going on. Does this apply to autoimmune patients? That's obviously a question that is very important and needs to be addressed. With that, I thank you for your attention and would like to acknowledge my mentors, my lab members, and all my funding sources. And I'm looking forward to questions after the second talk. Thank you.
Thank you, Christoph, a lot. So this was really a very, very nice presentation and also a very interesting outlook. According to our tradition, we will already proceed to the next speaker, and we will have a very interactive discussion at the very end. And with this, I will pass on to Remco, who will introduce the next speaker.
Thank you, Christina. Before introducing the next speaker, who is Ricardo Grieshaber-Bouyer, I would like to encourage the attendees in this live session to pose any questions they might have in the Q&A or the chat function in Zoom, so we can discuss them afterwards. So Professor Grieshaber-Bouyer studied medicine in Heidelberg. He did a postdoc with the Boston-based ImGen consortium, and then went back to Heidelberg as a clinician scientist. He's now a professor of clinical systems immunology at Erlangen-Nuremberg, and he has the clinical trial unit focusing on the interdisciplinary care of patients with rheumatic and immune-mediated diseases. Together with George Schett from Erlangen, he also explores the power of CAR T cells and T cell engagers for the treatment of autoimmune diseases. Please, Ricardo.
Yeah, thank you so much for the kind invitation and introduction to our work. It's a real pleasure to present here a little bit on our lessons learned from CAR T cell therapy and also T cell engagers. And I think that this is a very exciting time where we are really in translational immunology. Probably is the best time ever to learn things from the patients and also uncover new mechanisms, take them back to the lab. And so what we have already heard wonderfully from Christoph is that B cells are kind of getting another renaissance as a therapeutic target. They are getting now into the focus of autoimmune disease therapy, most likely because we have better ways to approach this challenge than ever. So we don't just have depleting antibodies, but we now also have cellular therapies and T cell engaging therapies. And so what we have witnessed over the past couple of years, so the first patient was treated in 2020. The first paper was published in 2021. So since then, we have a little bit more than 4 years now that this has been going on. We've seen really remarkable treatment success in treating heavily refractory patients that have systemic lupus, but also myositis, and even systemic sclerosis with lung involvement. And I just want to give you a little bit an overview of where things currently stand. So there's different categories of therapeutics that you can think about, and I like to group them in cell therapies and peptide-based therapeutics. So for the cell therapies, they are living drugs that bring their effector mechanism essentially for target cell clearance. For the peptide-based therapeutics, you're looping in additional factors that are already present in the patient's immune system, whether that's phagocytes, NK cells, or complement for the canonical monoclonals or T cells for the TCEs. And here for the cell therapies, what I like to point out, most data that we have to date is from autologous CD19 CAR T cell therapy. So that's the bread and butter CAR T cell therapy being used. And there's different variations with different co-stimulatory domains. But so far, there is not that much differentiation, one has to say. And this is an approach that works really well for depleting CD19 expressing B cells. But there is now lots of exciting developments also around allogeneic approaches to shorten the time between treatment decision and application. And what I want to dive in a little bit is this opportunity that we have to learn how these therapies actually work. So one thing that was postulated but so far not shown is that CAR T cells actually lead to a more complete depletion of B cells, and that this could be related to superior treatment responses in malignancy, but also in autoimmune disease. And as we can see here from a longitudinal biopsy study before CAR T cell therapy, after CAR T, and after rituximab as a comparator, looking at the inguinal lymph node, we can see that CAR T cells empty the lymph node of CD19 expressing B cells, unlike rituximab. So rituximab does have issues in depleting B cells completely in the tissue. What we also see after the B cells start recovering is some very interesting biology, because we can basically start to understand how the B cell system evolves in adults if you reboot it. And probably the lowest hanging fruit was first to look at B cell maturation stages, and you can see here that transitional B cells are expanded after they recover. If we look at the heavy chain, in the pre-CAR T patients, you can sequence a lot of class-switched heavy chains, including A and IgG. In the after CAR T recovered B cells, you only sequence the mu chain and the delta chain. So you really have only a naive repertoire here after deeply depleting the B cells. And what's quite interesting is that if we survey the B cell compartment over prolonged periods of time, even long-term surveillance over 1 year, and in many patients even over 1.5 years, we see that naive B cells stay remarkably high, and that the class-switched memory B cells stay at a very low level, even for extended periods of time. Also very important in the context of, in particular, systemic lupus, is a population of B cells that is called double negative 2 B cells. These are implied in driving potent extrafollicular responses to autoantigens and differentiate into plasmablasts that release autoantibodies. And these double negative 2 B cells are typically characterized by absent expression of CXCR5, which would otherwise draw them into lymphoid structures, and by high expression of CD11C and also TBET. And as we can see here, after CAR T cell therapy, this population of CXCR5 negative TBET bright CD11C expressing cells is absent, and we have longitudinally seen that it does not recover. Concomitantly, we also see a sustained reduction in autoantibody producing plasmablasts and also in activated plasmablasts compared to patients with active SLE, and this remains at a level of healthy donors. But it doesn't stop, interestingly, with B cells, because we can understand using different approaches also the impacts on non-B cell lineages. And so here, using the Verily profiler, which is a very comprehensive platform to look at sorted subsets from PBMCs, we were able to sort here different populations of monocytes, dendritic cells, CD4s, CD8s, gamma deltas, and K cells, and different B cell subsets, because we really wanted to have this systems immunology perspective on understanding immune reset. And as you can see, there is a substantial proportion of differentially accessible regions, differentially expressed genes, and also differentially expressed proteins that characterize the immune landscape after CAR T cell therapy. If we look at gene expression, for instance, we can see that we have a strong downregulation of DNA binding related terms across different T cell subsets. We also see a strong reduction in type 1 interferon signaling in effector T cells. And we see a reduction in neutrophil degranulation in the monocyte term, suggesting that we have a reduced presence of low density granulocytes. And this is indeed the case when we look here at the protein level as well. On monocytes, we can see in active autoimmune disease, we have bright expression of Siglec-1, that's an interferon marker on monocytes, but also CD15, which is coming from the contaminating granulocytes. And this is strongly downregulated after CAR T, implying that we have less low-density granulocytes. When we do profile the low-density granulocytes via single-cell RNA-seq, we see that they also have a reduced expression of type 1 mediated signaling, and they have reduced expression also of the anti-apoptotic BCL2, which shows us that we have reprogramming of neutrophils with decrease in effector molecules. We also see changes in the T cell compartment. For instance, here we look at the regulatory T cells, and we see before CAR T cell therapy, we have a lower expression actually of suppression-related markers such as PD-L1 and CTLA-4 in active autoimmune disease. And this normalizes to the same levels than healthy donors after CAR T cell therapy. So whether that is related actually to this to the reduced stimulus from pathogenic B cells, or whether this is also related to the expansion of CAR T cells in the system is not entirely clear, but it's very consistent that we have reprogramming in the T cell compartment as well. So what we learn from this is actually that T cells are very likely more potent effectors than canonical approaches, that we are now in a stage where we can realize ultra deep B cell depletion, and this is very likely leading also to superior clinical responses, potentially even with a curative potential. And we have validated new targets now, including CD19 and BCMA for autoimmune disease, conquered new autoimmune diseases such as myasthenia gravis also for B cell targeting via CAR T cells, and we're learning new mechanistic insights. And so the interesting thing is, where do we go from here? I think we're still continuing to learn a lot. One thing, for instance, is how do we manage disease progression? So we recently had a patient with JO1 associated myositis who relapsed after CAR T. As you can see here, we gave CAR T cells. Initially, the CK went down, that's the creatine kinase, and the muscle strength normalized in this patient, but then the patient relapsed. And what we did was actually to give the cryopreserved product after full dose lymphodepletion. But as you can see here, the CK continued to increase and we had no improvement in muscle strength. So what was going on? The CAR T cells were not expanding at all. You can see this here in purple. It's a flat line. And the B cells were just getting depressed a little bit via the lymphodepletion, but they were not reaching zero as they should if the CAR T cell product works. And the lymphodepletion had normal effects on circulating leukocyte counts with the same decrease, also in the same decrease in granulocytes. So we looked a little bit at what was going on, and interestingly, we didn't see any anti-drug antibodies. So here's the first CAR T therapy, no anti-drug antibodies. Second CAR T, no anti-drug antibodies. But we did find a T cell response. So remember, these the CAR sequences are highly proprietary. We were using an approved drug called Abecma, which is a BCMA. We were using before this the CD19 Miltenyi CAR. They don't release the sequence, of course, but we can test a peptide library that spans the entire CAR sequence. And here we could see a specific T cell response. If you look here at patients that get redosed with cryopreserved product, you can see expansion. And in this patient, for instance, there is no T cell response against the CAR. So it's very likely that this anti-CAR T cell response led to immediate rejection. And interestingly, over time, after this booster dose here of the antigen, the patient then later developed also anti-drug antibodies in the context of T cell help and in the context of insufficient B cell depletion. So then we bridged with daratumumab, which is a CD38 antibody. This brought temporary relief, but ultimately the patient progressed again, and we applied rescue therapy with the BCMA CAR T cell therapy, which led to complete B cell depletion, not just in the blood, but also in the tissues. and led to a complete response. Again, the patient is now 1 year treatment free. So what we learn from this important observation is actually that the lymphodepletion alone is not efficacious, that there are immunogenicity challenges that we need to think about and address that might hamper reapplication of the same CAR product, and that switching product and target can be a useful concept in the case of relapse. And I think, unfortunately, due to time constraints, we cannot dive into TCEs that much, but this is also very exciting arena, especially because it will enable off-the-shelf and outpatient application of B-cell depleting approaches. And so we are at this interesting stage where we have a convergence here really of autoimmune and oncology, having proof of principle for monoclonals, but also for CAR Ts and for TCEs. And so with that, I come to an end. Thanks for your attention. Happy to take questions.
Yeah, thank you, Ricardo, for this very nice talk. So we can now start with the discussion. So as Remco already mentioned, please feel free to type in your questions in the Q&A chat section, or just raise your hands if you like. So, and we already have the first question here. It might be directed to both speakers, but maybe I will ask Christoph to answer it, and not both for time reasons. And the first question is, what's the latest research on CAR T cells in pemphigus vulgaris?
Yeah, that's a great question. I think there are ongoing trials targeting CD19, recruiting patients, pemphigus patients. It's as was discussed, the question is, what is the correct dose? Do we need lymphodepletion? How long do you follow up to make the decision that you see basically complete remission? What are your biomarkers? I think pemphigus is good because you can always look at the autoantibodies that are disease-causing, the desmoglein 3 antibodies. And all of this was basically enabled by studies in lupus patients. Do we know if pemphigus acts like lupus? Will we see the same responses? Unclear. And but we're fairly confident that it should at least be, let's say, safe in a context that it's for refractory patients, something that is a good option. But we have to see the efficacy first. The AA CAR basically also showed that immunotherapy with a CAR T cell product is safe in autoimmune patients. 10 years ago, it was basically a contraindication. When you had autoimmune disease as a cancer patient, you couldn't get CAR T cells because people were concerned that these activated CAR T cells would drive autoimmune disease. And the efficacy of the AA CAR was not as good, or there was no clear benefit, clinical benefit. That's why a CD19 approach right now is being tested in clinical trials.
Okay, thank you very much. Yeah, and we have another question from Attilas Zölösi. You want to ask your question?
Seems as if Christina Zielinski is freezing. Perhaps I can jump in with a question to both of you, but maybe primarily to Ricardo. What do you suspect or expect will be the place of CAR T cell therapy for autoimmune skin disease in 10 years' time? Will this be a very niche type of ultra-resistant patients, and in particular as kind of compared to T cell engager and other therapeutic antibody approaches?
So the way I view it is, right now it's reserved for very niche applications, but I think this will change very, very rapidly and with exponential speed. And I think that which modality ultimately will be able to reach and benefit the most patients is still unclear. But there is a lot of technology development going on to make cellular therapies safer, more efficacious, more scalable, available off the shelf with less lag time. Then you have peptide-based therapeutics, which also in hematological malignancies can come very, very close and be perhaps even on par with cellular therapies, and which are, of course, a fraction of the manufacturing cost. Then you have in vivo approaches, which are coming very, very fast. We are just starting to see the first in vivo data coming out. But so far it's looking pretty good. And this field is not mature at all, right? So basically everything that we are doing now, we learn a lot, and from year to year, we don't have optimized treatment protocols. All the 19 or BCMA CARs and also TCEs had been developed for hemato-oncology. We don't really have dedicated autoimmune products yet, but all of this will come. And then, of course, for many skin diseases, you can envision also antigen-specific therapy, which is going to be another really big field. So my optimistic view actually is that in 10 years, this is the standard of care and it's very safe and we apply it very early on.
Do you agree, Christoph?
Yeah, I think I would agree with that. I do think, for example, if we take an autoimmune blistering disease, pemphigus, rituximab is FDA approved and works pretty well. The question is, what do you add in addition with the CAR T cell? And it would really be reduce the number of relapses, maybe even induce long-term remission. I don't want to use the word cure to,
Why not?
generously here, but it could be, definitely could be potentially curative if you look at some of the lupus data that have been published where patients are in complete remission for years and don't seem to have any clinical activity. So that would be really, where I would see that we justify using CAR T cells first line. If we have solid data that it really changes dramatically the long-term outcome, that patients don't have relapses, don't need to be treated, and are maybe even considered cured at one point. That would be the dream, obviously. And I think CAR T cells and any of these technologies, in vivo engineering or T cell engagers, may guide us there, and we just have to figure out how to safely do it.
Yeah, maybe to continue along this debate of whether they are secure or not a cure, I think we're getting a little bit into a debate mood. I was also wondering if the CAR T cells are supposed to also induce a complete immune reset. Maybe a cure. What about the longevity and maintenance of the CAR T cells? I think you showed, Christoph, a plot at the beginning that the CAR T cells are maintained long term, but where do people actually look? Do they just look in the blood and in the bone marrow? Is it possible that CAR T cells might be sequestered in certain tissues, and can they be retrieved? Again, what do we know about where they settle and reside long term?
Yeah, I mean, it's a great question. In cancer patients, it's known that you need the persistence of the CAR T cells for at least a couple of months to induce remission. In autoimmune patients, in lupus at least, doesn't seem to be the case. You can actually get shorter duration of B cell depletion if it's just deep enough. It's more about the quality of B cell depletion to induce this reset. And then the question is every autoimmune disease like lupus? Or do you need in certain autoimmune diseases long-lived CAR T cells? And you certainly can detect them in all tissues, at least in cancer patients. You can detect them in the CSF, you can detect them in the skin. The question is that good or bad? If you have a disease that tends to relapse, having continued immunosurveillance might be good if you're just specific enough. Yeah, that's certainly something that we have to figure out. And then all that oncogenic risk, if it's just a short-term, let's say, a CAR T cell that's not as long-lived, that is gone within a couple of months, that wouldn't even apply. But we don't know what disease requires what longevity.
Okay, and Ricardo, do you agree? Because I think you're also a specialist in this tissue residency issues, and maybe to continue along the line, are maybe also the target cells, the B cells, sequestered and dormant in some areas, because we always consider them as circulating cells, or do you think they are also pathogenic tissue resident B cells, maybe sleeping in the bone marrow, which are more difficult, not so accessible?
Right. So you're basically getting at the important resistance mechanism, right? What was shown in a very elegant paper a long time ago, it's a 20-year-old GI paper, is that rituximab actually reaches B cells and tissues, but doesn't deplete there. And the dynamics of B cell egress into vasculature, so really vascular access of opsonized B cells, was necessary to mediate their clearance. In fact, when mice were overloaded with rituximab literally every other day for like 4 months, and you still had B cells in tissues, but they all were stained with rituximab. And so the B cells that don't circulate a lot, especially in Peyer's patches, they are more resistant to depletion. And in some areas of the marginal zone or the spleen, these are the most resistant. But we also know from abdominal lymph node biopsies, for instance, that you don't have full depletion. Whereas in the blood, it's very easy, right? So in blood, you get to zero quite quickly. And so the idea that we have about persistence is that it's not so much about sticking around for a long time. It's more about really reaching something like a ground zero, a B cell free state. And then it can be 1 minute, right? But you have to completely clear and allow this rebooting without re-expansion of autoreactive clones. And then I think, the beauty is that if the autoantigens are still around and the naive B cells repopulate, then they become tolerant against these antigens, right? And so I think that's the vision for the long-term response.
Before closing the discussion, I would like your answer to a final question posed by one of the attendees, but that needs to be brief, although it might deserve a longer discussion. That is, what is your experience and opinion about CAR Treg cells?
Christoph, you want to start?
Yeah, I can start. I think it's certainly promising. It seems to be more challenging to generate them and make sure that they are actually Tregs in the long run. And there are promising preclinical data and some clinical studies that show that they're at least safe. I think it's certainly another avenue, another modality. Understanding human regulatory T cell biology and then trying to engineer that or re-engineer that, certainly almost a field on its own. It's not as simple. Yeah, that's my perspective.
Ricardo?
Yes, I think the identity issue is challenging for the Tregs, and then keeping that identity. But otherwise, I think the concept is very elegant because you don't necessarily have to deplete. And I think especially when you have diseases where you have well-known antigens where Tregs could tolerize the environment, and then you can essentially have a spreading of the tolerizing effect onto other antigens as well, is quite beautiful, right? So I'm looking at this actually with a lot of enthusiasm, but I think that, the persistence of the Treg phenotype is something that needs to be addressed.
Well, thank you very much. Also on behalf of Christina Zielinski, I'm afraid we need to close the discussion now and also the Q&A. Well, your presentations have really refined our ideas and also given a lot of promise to this new CAR T cell therapies for autoimmune skin diseases. I would like to finish this sweet and sour session by highlighting the upcoming webinars. So the next kitchen session will be prepared by Carmen Gerlach, which will be on August 27th. And the next sweet and sour session will be on November 12th. And this will be a debate about the harmful and beneficial effects of sunlight. We'll have to close there. I'm not sure if Christina, if you would like to add anything.
Please remember that we have a beautiful meeting soon in Antwerp, and you still have the chance to submit an abstract. And otherwise, I'm actually very happy about this webinar. It was very exciting. I would have loved to continue the discussion for a while, but we are restricted with time. So I really thank the speakers, and I also thank the audience for the questions and for being with us. And I'm looking very much forward to seeing you next time in November. Have a very nice day. Bye-bye.
Thank you so much. Have a good day. Bye.
Thank you. Good day. Bye.